all the leukemias are back there, and those are all real patient specimens too. There's not a store-bought thing in here, so you get to see the stuff that we get to see, and it's all dead. Relax. It won't hurt you. It may scare the daylights out of you.
I can't because it is.
As ugly as some of it is, but no, it's not.
Just remember.
So let's see what we got. And this is what I said about this being kind of hard to see. These are the normal types of blood cells that you're going to be seeing. The red blood cells in the background are these are all normal. They have a slightly clearer center to them because they're biconcave discs. These are only, there's a bunch of other abnormal ones that you're going to see too. These are, what? Come back here. There we go. This is a lymphocyte.
Can you move the screen closer to us?
Say what?
If it's on wheels, can you move it closer?
If we get him to move back a little bit.
Because it's very
You
small.
want to move back a little bit while we're watching this? Because it would be better a little closer. Of course, I got to move back too. Oh, thank you.
You wanted to try here.
Or you can pretend you can see.
Oh, that's good. Yeah, that's better.
That doesn't help much.
Okay.
Much better.
Thank you.
Sure.
Yeah, this is a lymphocyte. This is a lymphocyte, the kind we normally see in young people up to maybe 12 or 14. They're a little bit bigger. You see there's some indentations around some red cells there. This is a neutrophil. This is one of the scavenging cells that goes around and picks up stuff. Another one. These are segmented. Here's another version of it that's a little younger. It's kind of a horseshoe-shaped nucleus. Those are called bands.
It's a monocyte, right?
Huh? No. This one. Yeah, that one is a monocyte. This is the band I was talking about down here. That one is a monocyte. Good night. Don't ask me how I see. That is a monocyte right there.
That's a monocyte.
And another small lymphocyte. And this is a basophil down here.
Basophil.
I can't see these any better than you can. I was expecting a bigger screen, so these things would be big enough for everyone to look at. Now, when these things are in the bone marrow, which is where they're essentially born, as they mature, they get bigger and the coloration gets darker. You can see here that the size gets a little bigger as they move into cell division, and then once they divide, they start getting smaller.
Is this a erythrocyte?
Say?
Is this an erythrocyte?
Actually, they're all that way, to one degree or another. That is probably closer to red cells, erythrocytes.
Yeah.
Red cell changes, we end up with coloration starting at a dark blue and moving all the way down to the normal kind of salmon colored or pink color. This is the nucleus size. You can see it starts out pretty big and a little more diffuse and gets dense, and then it gets ejected from the red cells, so you end up with normal stuff there. They have, this is the nuclear concentration, for lack of a better word. It's more diffuse in the younger cells, and it kind of clumps up the older the cells get, and this is actually what they end up looking like. This is what's called pro-rubrocyte, I think. A rubrocyte, that's what they call the red cell group. And as they come down, you can see the nucleus get smaller and smaller and smaller, and then it's gone, the last couple of them.
Now, is this just in the bone marrow? Or
Primarily, yeah. It should be just in the bone marrow. You don't always see it just in the bone marrow, particularly the nucleated red cells. You can see those in circulation in a lot of
I'm just wondering, you're talking about maturation. I didn't know if that was
That's all
after
bone
it's been released into the blood.
No, it's primarily
So this is
bone marrow.
all-
Yeah, it's
okay.
primarily bone marrow. This is hard to see. This is the basic takeaway from this is that everything comes from one stem cell. All the red cells and all the white cells come from this one stem cell up here at the top. This is the monocytic version over here. When you get down to myelocytes, myelocytes are the main blood cells, the white cells. The same thing goes to basophils, regular white blood cells, and eosinophils, and then those mature down there. So they all come from the same thing. Platelets start out, they're called megakaryocytes when they first start out. They get bigger and bigger. Down here you can start to see little pieces of the cytoplasm. You guys okay back there? Little pieces of the cytoplasm break off. Platelets aren't cells, they're pieces of cells. And down here is what you get at the end. And boy, this is terrible. This is too small. These are the lymphocytic series, and don't worry about these. These are those are plasma cells. Don't worry about those. These are the red cells down this side.
But are platelet is just a part of the cell?
The myelocytic series, this is just a piece of the ones that we had there. You're gonna get to see most of these in the cells you're looking at back there. This is just a bigger version of what we just looked at. The basophils here, the regular myelocytes, down to the neutrophils here, and the eosinophils are here. These have dark blue granules. These have orange granules. Now, the basophils in some of the stains tend to be kind of water soluble, and so most of the granules end up getting washed out. So you'll see a few of the dark blue granules, but nowhere near as many as you see here most of the time. Every now and then you'll see one like this, but not always.
She had a question about the platelets.
Yes,
Oh, I didn't know it was a fragment of a cell.
yeah, yeah, they're a piece of cytoplasm. That's the best way to describe it.
Oh, interesting.
Yeah. And platelets are platelets are involved in coagulation. They're the base thing that shows up with fibrin to start the clotting mechanism.
They're the bricks.
Hmm?
Bricks. The bricks in the wall. Platelets.
Oh, I see what you're saying. Yeah, that's not a bad way to put it. The foundation of the structure, that and the fibrinogen that holds, the fiber that holds all the stuff.
Is that what the white cells are?
Hmm?
No.
Couldn't hear you.
Okay.
Yeah, these are white cells. These are monocytes. Oh, jeez. Get back here. And basically just presume all these are different kinds of monocytes. Because that's all we're really trying to do here. The lymphocytes range from very, very small to pretty good size where they're indented here, and you can see them back in the book you've got there. It's because they're not showing you the red cells there. The membranes of these things will actually fold around the red cell. That's one of the marks of them. They're just very soft, pliable membranes on some of them. Okay, this is going to be neutrophils here on the left side, neutrophilic granulocytes, monocytes down the center, and lymphocytes on the right side. So you can see that until you start looking at these things really close, they look an awful lot alike. And when you get to the younger ones, the blast cells that you're going to see a bunch of, trying to tell them apart, you end up telling them apart by the company they keep, essentially. If you see a lot of neutrophils, it's probably a myeloblast. If you see a lot of lymphocytes, it's probably a lymphocyte, or lymphoblast. If you see a lot of monocytes, it's going to be a monoblast, probably. Because remember, they all come from the same stem cell, and if they're young enough that they're close to that stem cell in maturation, there's not going to be any defining properties that you can use to tell them apart. We have to try to tell them apart anyway. Now, when I was doing this, the use of special stains and fluorescent things and that kind of labels for surface proteins were kind of rare. You didn't do them all the time. A lot of the time the morphology was what you based everything on. Now they'll send these things out for special stains and fluorescence, fluorescence in situ hybridization fish and a couple other techniques so they can identify them all the way to B cells and T cells and that sort of thing. It's much more accurate than looking through a microscope. Blast progression, you can see they start big and they get smaller. The monocytic ones will start to form lobes, and that's actually plasma cells. You're not going to see very many plasma cells. The mark of a plasma cell is kind of this clearing area next to the. I can't see very well either. We were supposed to have a big screen.
Sorry, all the theater's in use.
Well, art has its moments.
Yeah.
I thought we were going to have a big screen. I've done this differently. This is the megakaryocytes, the platelets. Just take away from this the big ones. You're not going to see very many big ones, unless if you look at a bone marrow, you'll see them. If you're not looking at bone marrows, they'll be pretty rare. But the platelets are down here. You'll see large platelets sometimes, a little funny looking, but they're just coming out in a hurry because there's a bleeding process or the blood's being used up somehow. Because as you use these things up, more get produced to try to backfill everything. That makes sense?
Yeah.
This is the same thing with the red cells. You just see the progression as they get smaller and smaller. You'll see a whole bunch of those things. Particularly, all of these things you'll see in the bone marrows. And there's probably a dozen bone marrow slides back there. Some of them are malignant, some of them are normal. Here's red cells that have pathologies of one kind or another. Don't worry about these. You may see some of the things, but that's just some of the things that you run into. These big ones come up back here. These big ones, you almost never see unless you're looking at somebody with a specific disease. You'll see that this is a malarial specimen. You probably can't see it back there at all. There's some malaria slides back there. This is called a ring form. These others are just younger ones. Red cells in disease states can have all kinds of different shapes. They can get broken up. Sometimes you have genetic problems that cause them. You can have G6PD deficiency, you can have vitamin D deficiency, you can have all kinds of things. Normal ones are up here in the corner. These are what they call target cells because they look like targets. A lot of sickle cell patients have target cells, thalassemia patients, all kinds of things. These are spherocytes. There's some spherocyte cells back there. They don't have that central pallor to the red cell. They're dark, and you can tell when you look at the slide, they actually look more like spheres than normal red cells. You probably won't run into any of these up here. You will see some sickles. These are sickle cells, although these are really much more dramatic than you normally get. You normally get ones that are a little bit twisted and kind of pointed. There is a couple hemoglobin C, SC slides back there. You get hemoglobin C crystals along with the hemoglobin S, and they actually form crystals inside the red blood cells, so you can see they're funny shapes, and you can actually sometimes see what looks like long white crystals now. That's probably about all we need to do with that. Oh, and some valocytes. There's a couple of slides in there of hereditary valocytosis, so all the red cells that are normally round are oval. It's kind of cool looking, it doesn't do much. There's some occlusions that you see. Don't worry about these. It's just trying to see them from back there. If you see things, a lot of the ones that you see bigger dark blue chunks in there, they're just remnants of the nuclear material. Here's this is an iron deficiency anemia. I don't know if you can see it very well, but there's some teardrop shaped cells, and they're really what they call hyperchromic. There's nowhere near as much hemoglobin in the cell as normal cells. These are normal in here. Pernicious anemia, I think there's 2 pH slides in there. These are bigger and denser. This is some of the ovalocytes, and this is a genetic condition called thalassemia, where there's all kinds of stuff, target cells and all kinds of other junk.
Can you give us just a definition of hemoglobin?
It's the molecule that carries the oxygen.
Thank you. In the blood, obviously.
It's a
Unless you've been
combination,
cut, in which case it's going to be something.
it's a
someplace else.
combination of
Yes.
heme and iron, and it forms a, it attaches to oxygen
Okay.
and carries it through the blood. Yeah.
If you'd like, I can send you
Yeah, let's do it.
the,
Yeah.
yeah.
Yeah, it's a,
And it's the oxygen actually that makes the hemoglobin red too.
We have another question from Henry.
Red or, okay, there's going to be several slides back. There's a bunch of them on this. The only ones you're going to have to worry about are the left 2 columns. I've never seen, well, let's see, this is Plasmodium falciparum on this column here. That's the most dangerous malaria around. This one kills most of the people that are killed by malaria in the world. This one's Plasmodium vivax. There's a bunch of vivax slides back there. I think there's 3 or 4 falciparum slides. Vivax is the one you see mostly in the United States and in the Americas and Southeast Asia. Falciparum is mostly in Africa. We get a lot of travelers with it now. And these other 2, malaria and ovale, I've never seen. In 40 years, I've never seen one, so I'm not even sure where those are.
have you seen the trophozoites?
Huh?
Have you seen the trophozoites in blood springs?
They all have tropes. Yeah, they all have tropes. But yeah, there's some, actually, there's a couple slides back there that have these banana-shaped macrogametocytes. The only thing that's easy to tell, species-wise, on these things is the if you see a banana-shaped macrogametocyte, and some of those slides have them, I checked them yesterday, then you know it's Plasmodium falciparum. The rest of them takes a great deal more work and painstaking staining and a lot of stuff to figure out which they are. They look very, very much alike. What else we got here? Okay, this is, anybody know anybody with lupus erythematosus? Lupus? Okay, in lupus you end up building antibodies to your own white blood cells. And sometimes circulating, you'll find white blood cells that because of the antibodies have started to disintegrate, and you'll see essentially just a purple blob. It's purple because we've stained it. And then other white cells, neutrophils, will come along and actually phagocytize it to try to pull it out of the circulation. And what this is actually a test we used to do called a LE prep, where we kind of force it to do that by just the couple of tricks we have, rocking it back and forth and incubating it at room temperature for about 8 hours. But these things in the center, those are the nucleus of red or white blood cells that have been destroyed essentially by the antibodies, and they've been captured by the others to pull out from the.
So that's numerous cells. That's a number of cells. Each one of those is more than one cell. That's a
Yeah,
number.
well, it's one cell now, but it has the remains of other cells in it. This one's got 2 other cells in it. That's got 1, 2. That's got 1. That's got 1. That looks like it's only got 3. But yeah, they're being eaten essentially. It's kind of a cleanup brew. Infectious mononucleosis. Infectious mononucleosis is kind of, it's different. You'd think that it's all monocytes by the name. But mostly it's all lymphocytes, except the lymphocytes all look like monocytes. These 2 cells right here are monocytes. All the rest of them are lymphocytes, even though they look like more like monocytes than lymphocytes. That is actually, if you have a really good case, it's not that hard to find. If you have one that's kind of on the edge, it can be kind of tough. Yeah,
What's the difference between a monocyte and a lymphocyte?
they're different cells.
Thank you for asking.
Yes, but I mean,
Look-wise,
No,
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 what, it would be
What's
really
the
helpful.
purpose?
oh, the purpose, lymphocytes
There
are,
are some people that we don't know the basics of all of this.
yeah, okay, I should have said that.
So
Lymphocytes,
just give like a general definition.
yeah,
of-
lymphocytes are where, for better or worse, your antibodies are gonna come from, the very
Okay.
types of lymphocytes.
Lymphocytes.
Monocytes are scavengers like neutrophils.
Okay.
They're macrophages essentially. They actually move around. They're closer to amoeba than the neutrophils. The neutrophils can move too, but the monos move quite a bit. And so they'll go surround something and eat it. The eosinophils are reactions to let's see.
Okay, and how did they compare to a red blood cell?
Oh, they're totally different. That's like comparing your hand and the top of your head.
How do they look?
How do they look? You've seen big lymphocytes and red blood cells? That's what we went back through in the beginning. That's a red blood cell.
Okay.
There's no nucleus in red blood cells except very young ones.
Didn't know that. That's interesting.
There's no nucleus in a red blood cell?
No, not after they've come out of the bone marrow. There's nucleated red blood cells in the bone marrow. They start with a nucleus and they extrude the nucleus as they go out through holes in the bone marrow to get out into circulation. In babies, you can have lots of, particularly if they're sick, you can have preemies, you might have, for every 100 white blood cells, I've literally seen 600 nucleated red blood cells, which is pretty insane.
Okay,
You probably won't have any.
I'm sorry to ask this very basic question, but if you prick your finger and a little blood comes out, and it's usually red, and I'm assuming it's a red blood cell,
That's all the cells.
and is it?
The red is the
right,
hemoglobin,
right.
yeah.
Is it gonna be a nucleus in that?
Shouldn't be.
No, okay.
Now, if you have some nasty disease going, there may be a few. All right, where were we?
The eosinophils are typical when you have allergies.
Yeah, they're allergies. I forgot what allergy.
I knew, that's what I was waiting for.
Basophils, we're not really certain what basophils are supposed to be doing. They seem to have some functions. The one thing that we're not sure why it works either is if you find a fairly large number of basophils, and the number is usually less than 1 out of 100, it's less than 1%. If you find a fairly large number of basophils, 4, 5, 6% or more, you better start looking for a tumor because it's highly likely that the patient has cancer somewhere. We have no clue that I know of why that's the case, but it is. I suppose there are times when you won't find a tumor, but there's probably something there somewhere.
When blood gets sent off to a lab for a metabolic screen, when they're doing the micros on that stuff, are they going to be able to see this stuff, or do you have to look for it specifically?
Yeah, well, a metabolic screen, if it's got a CBC with it, they're gonna be doing this. If it's just a metabolic screen, a basic metabolic panel, it's all chemistry. Metabolic panels don't include this. This is gonna be in CBC. And they should be seeing these things if they're there.
Okay.
Now, most of the analysis is done by automated analyzer. And instead of counting 100 cells, like we used to do, and assigning a percentage, if you find 2 cells, it's 2% out of 100 cells. You get a rough idea how accurate that is. It's accurate enough for diagnostic purposes, but it's not as accurate as it could be. The newer analyzers are going to count 10,000 if there's enough cells for it to count. So they're going to be a little more accurate like that. They will miss some of the strange stuff. Let's see, where were we? Okay. This is an interesting, Chediak Higashi. This is something, they're according to UCLA, there's less than 200 of these cases, ever, worldwide. The only reason I put this up here is because back there I have 5 slides from Chediak Higashi, because where I was working at Arrowhead in San Diego County, we had a patient. And I made some extra slides. So you can see this, it's a genetic error. Most of the patients die by the time they're 10. And you end up with, it's a liposome issue of some kind. I don't remember exactly what. But you end up with some strange inclusions around the white cells, these dark inclusions. And those slides actually have them, you can see. Other than that, most of us are told about these things or taught about these things, and nobody ever gets to see it because they're so darn rare. And it's listed in those books back there. And actually, this is one of the pages of it. And I was lucky enough to get to see one, so I thought I'd toss that in.
Your luck is somebody else's bad luck.
Oh, Jim,
Yeah.
that's so true.
These are some parasites. The one on the left side here is, this is actually a yeast form called histoplasma. It's not very common here in Southern California. This is mostly Ohio River Valley. This is one that you don't see around here very often called leishmania. I don't have any leishmania slides. I've got several slides of histo, but they're not in this kind of macrophage. They're actually internal in neutrophils. So you can see those. Those are pretty rare too. Okay, normal white blood cells. So you know what they look like. This is going to be a neutrophil. Can you see these? Or am I on your way?
No, you're not.
Okay. This is a neutrophil. You can see it's got a dark, the chromatin of the nucleus is condensed. It's lobulated. It can have anywhere from 3 at minimum, usually 4 to 5 lobes on it. Those lobes should have little pinched areas between them. If they're not completely pinched off, then it's a little bit younger, and we end up calling them, those are the bands that I was talking about earlier.
How common do they call them polys?
All the time.
All the time?
That is
They're
just...
polymorphonuclear.
Yes, that's what PMN stands for, which is the other thing they call these things. Yeah, there's a lot of names depending on where you're working or what you're looking at or what book you're reading, because somebody else came up with a name for it. Yeah, these are what they call polys, and it's because the polymorphonuclear nucleus. This is a normal adult lymphocyte, nice kind of a tight round nucleus with not a lot of cytoplasm around it. The one thing about lymphocytes, the small ones like this, if you look at the cytoplasm, it kind of looks like somebody drew it and took an eraser to it and smudged it out. They just looked a little bit softer like that. And any cytoplasm, come back here, wrong button. Any cytoplasm is going to be primarily blue of one shade or another. Monocytes have the cytoplasm, there's granules in it. There's very few granules in lymphocytes. There's granules in this that are very, very small. They refer to it as a ground glass appearance. But the granules and the cytoplasm kind of are gray-pink, okay? Eosinophils, because they take up the eosin and the stain, are orange. All these granules here are kind of an orange-pink or salmon color. Most of the nucleus are bilobe. They're just 2 lobes. There's not 3 or 4 like there would be in some of the other cells. Basophils look a lot like eosinophils, except, come back here, except the granules are blue-purple. And like I said, most of the ones you're going to see don't have anywhere near that many granules in them. Sir?
About how big are these cells?
The basophils and the eosinophils are the same size as the neutrophils. Monocytes tend to be a little bit bigger. Lymphocytes tend to be smaller.
You can see the
In terms of
adult.
like microns,
Yeah.
would you say?
Well,
Yeah, for
a red blood cell, the average diameter of a red blood cell is 7 microns. So you can see in comparison, these are all normal. So lymphocytes are a little bigger. They're probably 10, somewhere in there. But some of the bigger lymphocytes can get pretty good size. You're going to see all kinds of things on those slides.
Now this looks like right stain.
Yes.
Type of stain.
Yeah, the right stain is the normal one that's being used. If you're trying to look at some of the malarias, you're going to be using GIMSA because that's the only one that's going to show a couple of the morphological things. Most places are using modified right GIMSA. I couldn't tell you how it's modified because every manufacturer has their own proprietary stain. The one common one I don't like so much now. It looks great on all these normal things, but some of the malignancies, if you've got myeloblasts. Some of them have what they call hour rods, or a little pink rod in there that will just instantly allow you to diagnose it. Problem is, some of the stains don't show hour rods very well. The ones we were using at Seminole County, I rarely saw hour rods, and I know there are more hour rods there than that. So
The cells around the white cells, are those red cells?
yes,
Okay.
yeah, these are all normal red cells.
Is the most common stain still H&E, hematoxin, and eosin?
For tissue, yes. For this stuff, Wright's is a combination of, depending on which one, eosin and methylene blue is not hematoxin. But there's some variations, and in Beams has got some other stuff, and I'm not certain which is which, because they don't really do a very good job of telling you, unless you're spending money for a specific stain. You just buy the reagents that come with the analyzer and plug it in, turn it on, that's what you get. We're lucky they make slides for us now. Okay, most of these red blood cells are perfectly normal. The only problem, you see these little teeny pieces. We get something like that down in the laboratory, we don't even have to call the emergency room and ask them what's going on. It's a burn patient. What's happening, as the blood's flowing through the capillaries in the skin, and the skin is burning and the capillaries burn as well, and as things start to collapse and close off, the red blood cells break up as they're trying to go through there. So this
How would
is
you differentiate those from platelets?
platelets are going to be blue-gray, and they're smaller. Actually, you might have a hard time, the really small ones, except the color is going to be dramatically different. There actually are no visible platelets in. Well, no, this is a platelet here. There's a little platelet there. They're kind of a bluish purple. But it's easy with pieces of red cell if you're actually looking at them under the microscope to tell the difference. It's a little harder on the screen here. Sickle cell. Remember I told you that the sickle cells aren't as dramatic as that diagram was showing. This is a sickle cell here. You can see just a little crescent. This is a sickle cell, even though it looks more oval. Here's another crescent that's a sickle cell here, and another one here. And that's about as good as you get. There's another one down here. But you see there's some target cells in here. There's a couple other pieces that are broken up in places. Sickle cell, most of the time you see a patient with sickle cell disease, unless they're in a crisis, you're not going to see a lot of sickle cells. And unless it's really loaded with sickle cells, most laboratories are going to have to do literally a test for sickle cell for hemoglobin S before they'll turn them out, because there's other things that can do this. And if you turn out a test that says somebody has sickle cell disease and they don't, that's not going to be a very nice thing. They're not going to be very happy with you. Okay, malaria. You can see how hard these are to find. Up here in the left-hand corner, upper left, is a ring form, like the growth progression that I showed you earlier in these things. You're going to see a whole lot of different shapes in here, and they're really difficult to tell which is which. There's another malarial parasite down here. These are amoeboid parasites. They're all mosquito-borne. You don't want that. This is the one that'll kill you. This is Plasmodium falciparum. One of the things that it does, you get multiple infections, so you might find two rings, like here in the cells. Here's a ring, here's another ring. These are hard to see at 40x. I can see them because I know what I'm looking at. You're probably going to have to find them on oil. And another one up here. The banana-shaped macrograminocytes I was talking about are right here. This is actually, these form inside red blood cells. So there's times like here where you're going to see the red cell with the macrograminocyte in it, the malarial parasite, and it's stretching the membranes on the red cell. It hasn't broken it yet. Eventually it breaks out.
There's a neutrophil off to the left.
Yeah, this is neutrophil. Actually, this would probably be called a neutrophil, because you probably assume that it was pinched off back here. But remember I was mentioning the pinching off between band forms and the older neutrophils? If they just look like this, you'd call this a band. And there's places that would call that a band because you couldn't see anything pinched off. Does it make much difference? No. Same cell. This is African sleeping sickness. I haven't heard much about this lately, but this kills a lot of people in Africa. These are this is Trypanosoma brucei. These are trypanosomes that are in the blood. This is transmitted by the tsetse fly. I haven't heard much about that lately either, but it kills a lot of people every year in Africa. Anytime you see this in the United States, it's because it came in on a jet. And you see a lot of stuff that is isolated in places now that people can get from the middle of the Congo or any place else and show up in your hospital in 18 months or 18 hours. So all the infectious diseases that used to be limited to places out in the weeds aren't limited to places in the weeds anymore. That's where they mostly are. But you can see them show up in your emergency room, show up in your office, show up in your neighborhood. They can walk into the house you got next door.
Forgive my ignorance, but are those bacteria then?
I'm sorry?
Are they bacteria?
No,
No, these are protozoa.
they're,
It's trypanosome.
they're flagellate.
Yeah. Yeah, and actually, you can see a little membrane, this little reddish pink line along the outside of them. That's how they swim, is by moving that undulating membrane. This is Chagas disease. This is the same kind of thing, but was primarily in the Americas, in South America and Central America and Mexico. The problem is it's now endemic in the United States, and hardly anybody knows about it.
Is that from the kissing bug?
Say again?
Is it from the kissing bug?
Yes, tritoma. Yeah,
Do you get
yeah.
one
The
bug
kissing
from-
the kissing bug's this little bug, and they call it a kissing bug because it tends to bite you near the lips.
You get a lot more than that.
But
Okay,
it looks like.
and the problem is, as soon as it gets its blood meal, it has the just lovely tendency, you may know people that do this, that it proceeds to take a crap right after it bites
What are
you.
the smaller purple dots here in the previous slide too?
Platelets. Those are platelets, they're just stained dark.
What?
Platelets.
Okay.
Those are the little things that help in clotting. Now, the problem with that is it itches after they bite, so you reach up to scratch the itch, and you take all the triatoma poop that is loaded with these things, and you push it into the wound they created by biting you, and you just auto-inoculated yourself. Not a lot of fun, but the interesting thing, UCLA just came out and said that they believe that there's 45,000 cases of this in Los Angeles County, and 300,000 cases in the United States, mostly down along the south. The bad thing about that is they presume that less than 2% of the people know they have it. And this actually causes heart problems. It causes issues with the heart muscle that has resulted in heart transplants in the past. So you don't want this. You really don't want this. This is chronic myelocytic. This is one of the leukemias that there's going to be a bunch of slides back there of.
So there are different kinds of leukemia?
Oh yeah. And none of them are fun.
Um,
If you have to get a leukemia, chronic lymphocytic leukemia is the one to get because it's relatively benign. Chronic myelocytic leukemia, it's chronic because it's not going to kill you very quickly. There's acute myelocytic leukemia, acute myelomonocytic leukemia, which is probably the same thing. Acute lymphocytic leukemia, there's a whole bunch of them, and they're just differentiated by how fast they're going to kill you, actually, and how rapidly the cells are being butted up. The thing with leukemias, all these cells that you're seeing, all the blast cells and the later cells, they're relatively normal-looking cells most of the time. They're just found in bone marrow, but they're being produced so rapidly that they get pushed out of the bone marrow to the point where you see them in the peripheral blood. So instead of having a white count that's somewhere between 5 and 10,000 per microliter, you might find 400 or 500. I've seen 800,000 in cases of acute myelocytic leukemia. Oh, I thought you were raising your hand. But this is a nucleated red blood cell. Okay, that shouldn't be here in peripheral blood. It's got pushed out with the rest of the stuff. This is a normal lymphocyte. This has been kind of stripped of its membrane. This is it's a blast of some kind. Here's another normal lymphocyte, and these are myeloblasts. There's a nucleoli in here. There's no granules in the cytoplasm. There's another myeloblast down here. This is a myeloblast. This is trying to distinguish myeloblasts from monoblasts from lymphoblasts, if they're really early, like we were saying earlier, is really, really hard. We basically don't do what they call myeloid leukemias now to avoid worrying about that.
And this,
There's a bunch of these slides back there, various kinds of these. And they all look different.
The size is also an issue?
Notice they're bigger,
Right,
they're much bigger.
right.
Blasts are gonna be much bigger than the later cells. Remember the progression I was talking about earlier, where they start big and get smaller, that's what's going on here. So the bigger the cell, the more likely it is it doesn't belong there. This is this is a panorama I took because it looked pretty. This is myelocytic leukemia. Here's some blasts. This is probably a normal band, a lymphocyte, another lymphocyte down here. Not too sure what that one is. That looks like a band. And here's another blast. Here's another band, lymphocyte. You can see there's lots of stuff. This is actually chronic, believe it or not. When the chronic ones, you end up with a lot of normal cells interspersed with the malignant ones. On the acute leukemia, stuff is being pushed out so fast that the cells don't have time to mature. So you don't see see very few normal ones. Here's acute myelomonocytic leukemia, so it's partially monocytes, it's probably partially myelocytes. Remember the hour rods I was mentioning? There's one right here and one right here. If you see an hour rod in one of these things, and I don't think you're going to see them, I don't think I put any slides back there with hour rods on. They're really, really hard to see, and it takes oil for the most part. But that's going to be diagnostic for myelomonocytic leukemia or myeloid leukemia. And you can see normal red blood cells. This is a little far into the smear, that's why they're looking like this. The farther you read into the smear, where things are a little more dense, there's more cells, they start looking kind of funny. So when you're looking at these things, you only look at the smear itself. Out where the red cells are really just about touching, maybe just barely touching themselves. Any farther in where they start overlapping, it gets too thick. So the cells you're looking at start to bunch up. Any farther out, they're fine. They just take a long time to find because there's not so many cells there. Lymphocytic leukemia. One of the things about lymphocytic leukemia, usually there's not a whole lot of cytoplasm around the nucleus. These are all lymphocytic blasts. There's a nucleoli in here. There's a nucleoli in this one. They're kind of hard to see. But notice how these are a little bit bluer. But this is a lymphocytic leukemia. These are normal red blood cells. This is a normal lymphocyte down here. See how much bigger they are. And it looks kind of like they smudged some of the chromatin out with a eraser. These are mesothelial cells. You've got endothelial cells, lining cells, essentially cavity lining cells. Endothelial cells are the very early layer, mesothelial cells are the middle layer, and epithelial cells are the outer. That's what you have on the surface of your body's epithelial cells. Well, this is from the middle layer. I show these normal ones because the ones that are come after this, a lot of them are malignant and they look different. They look like a bunch of different things. That's why there's a bunch of them here. But all of these are normal mesothelial cells. This is the way they normally look. Just a flat cell with a round nucleus and kind of a purplish stain to the stuff. There's usually an area actually where those arrows are, where there's a little bit of cytoplasm. Usually the nucleus doesn't quite go all the way up to the edge there.
This is not blood.
Then, no, this is going to be fluids. You're right.
Before you leave leukemias, does CLL look like anything in particular?
I have some CLL slides. Okay. CLL is going to have, I don't have very many of them. CLL has mostly normal looking lymphocytes. Normal, adult, small, there's just a lot of them. Mm-hmm. And if you get up to the 400,000 range, it's loaded. Usually they're 20 to 60 or something like that, much more than that, and they start causing issues. But no, they're just normal lymphocytes.
Yeah.
This is malignant mesothelial cells. I have no idea. This is a peritoneal fluid, abdominal fluid. I have no idea where this came from, what cancer was. But you can see the difference between the normal mesothelial cells and these. Huge nuclei, sometimes multinuclei. The normal ones can be multinucleate too. But you'll see they don't finish dividing when they multiply, so you can end up with them sticking together like this. And the cytoplasm can be very, very blue, lots of vacuoles in there. It doesn't say here, this is probably an adenocarcinoma. I say that this is lung cancer in chest fluid, peritoneal fluid, or pleural fluid, I'm sorry. I say it's probably adenocarcinoma because adeno has a tendency to not let the cells finish dividing, so you end up with big clumps of cells. This is a normal mesothelial cell here, this is a normal mesothelial cell. This is, actually that looks like a basophil. Those are normal neutrophils, but these are giant clumps of cancer. You see that, and yeah, it doesn't bode well for the patient, because these, when you see them in fluids, the cancer's already started to metastasize. That's the only reason the cells are in here. And once it's metastasized far enough that we start seeing the cells in the fluids, it gets very, very hard to stop. This is another one I thought was kind of cute. This is I have no idea what kind of cell. It kind of looks like a mesothelial cell, but it is, it's undergoing division right here. The chromosomes are broken apart. These are normal neutrophil or lymphocytes down here. That's a normal monocyte. There's a nucleated red blood cell. That's a normal neutrophil, segmented neutrophil, another lymphocyte. So you see how big that is. And anytime you see multiplication like that, anytime you see the chromosomes spreading apart, in fluids or in peripheral blood, you've got a problem. That doesn't happen under normal circumstances. This one was kind of cool. This looks almost like a normal mesothelial cell. This is in peritoneal fluid, but it has two nuclei, two nuclei. And the cool thing, this you can see here, the chromosomes are starting to break up a little bit, and they're way broken up on this one, so that you have two nuclei that aren't even using the same clock. It's asynchronous. Believe it or not, that picture's been hanging on the wall in the hematology lab I used to work at for about 15 years now, because they're pretty rare. This is an adenocarcinoma. You can see the division going on here. Shouldn't be seeing that. And here's a huge cell with multiple nucleuses in it, and see how ugly these things are. The mark of a cancer cell is they generally don't look very nice. Here's a normal lymphocyte down here for size. These are ovarian cancer cells. This is in a peritoneal fluid. This is an adenocarcinoma. You can see the lack of membranes here between the cells. It ended up looking kind of cool. This is a panorama I shot with like 33 frames of it. That panorama would be, I don't know, I think it's 13 feet long to be printed at normal resolution. The bad thing about it, the reason I did that was so I could photograph it under oil immersion and then back off, and so I can zoom in and out and see what it looked like. As a matter of fact, this is a slide that was shown in the Zoom meeting last week. Yeah.
So when the cells, the pathological cells are enlarged, the material are they still the same material that they normally would be? That is not like full
No.
of fluid?
Well, it's without going to genetically, no, they're not the same, because the genetic changes are making them look different. But what those differences are, I have no clue. That would take proteomics, and you'd have to go through and sequence a bunch of them and see what the protein differences were to tell what changes had been.
But it's not like they are filled with fluid.
It's not like they're still what?
Filled with fluid.
Still couldn't understand.
They still have
Filled
cellular cytoplasm inside.
the same.
Oh yeah, there is cytoplasm. What the cytoplasm has in it, its actual constituents, I don't know.
Okay.
That's an interesting question.
How did they make that slide?
That actually, that is literally a drop of fluid placed in one of these. This is a cytocentrifuge adapter. What you get, anybody got a slide? A slide goes on here. You put the drop of fluid in here, and then you have to go through and make sure it's diluted. Sometimes you have to put albumin in it to make sure it sticks properly. It can be a big drawn out thing. And you get it diluted enough that you have enough cells you can see, but not so many that they start clumping up. And then literally this is clamped up against a slide, and there's a special centrifuge, a cytocentrifuge that this goes in, that takes the fluid from here and spins it down onto the slide, and at the same time it spins it on the slide, this blotter paper here sucks up all the excess fluid, so that you actually get very nice looking stuff.
Because you're concentrated.
Yeah, well, it's not only concentrated, you get rid of all the excess liquid so that it doesn't ball up. Because if you're just trying to make a push smear like you normally do with blood or something else, you can't see this. It's really horrible.
And if you printed out, it would be 13 feet across? 13 feet across?
The print, yeah.
That's so intense.
Yeah, I mean, that's 33 good-sized frames in oil immersion. So, yeah, that was my first venture in panorama stuff. That was actually published in a pathology magazine in 2015.
Have you ever printed it out?
Huh?
Have you ever printed it out?
Not that big. But wait, I printed it, but not that big. I've been too lazy. The bad thing about this, as nice as that kind of looks like a flower arrangement. The bad thing about this is this ovarian cancer was in one of my tech's friends. And she lasted 6 months after we did this.
Okay.
That's the bad stuff about this stuff. This is not what we normally see in the hematology lab. This is actually a cytology prep. It's a pap smear, which is so you can see the nucleus anymore. But the shape of the cells and everything is the same thing we'd see. This is metastatic breast cancer in pleural fluid. And it's the same kind of clumps. It's probably an adenocarcinoma because of the way it's clumping. Same general idea. This is skin cancer cells in spinal fluid. This is metastatic melanoma, but you can see it's clumping up. You can see the individual nuclei in here. But this is a pap smear. They use pap smears because you can see the nuclear detail much better than you can in right stains.
What stain is this?
This is pap nickel.
How could it be from spinal fluid?
How do you get stick a needle in their back and on a syringe and pull?
No, I mean,
That's what metastatic means.
No.
It's not under skin anymore, it's somewhere
No,
else.
metastatic means it's metastasized.
no, I get that, but why, how could skin cancer be in spinal fluid and you say that it was?
Because it's metastasized. It's gotten inside. It's in the brain too.
I
Yeah.
see.
Yeah, once things start metastasizing, they can go everywhere. Anytime you have brain cancer that didn't start in the brain, it's metastasized and passed through that magical blood-brain barrier that most things aren't supposed to pass through. Yeah, it's pretty common. Here's a worm. This is Wucheria bancrofti. There's a couple slides back there of it. This causes elephantiasis, which is also known as lymphatic filariasis. This is mosquito-borne, I think. This is primarily Africa and down in Haiti and places like that. The bad thing about this is it causes that. It plugs up. It plugs up.
Okay, like leishmaniasis, the worms can bore into your skin if you're in the water. This doesn't
No,
have.
this is
mosquito
mosquito-borne. Yeah, this is mosquito-borne. And you can, I mean, the only way to treat this, you can kill the worms, but the only way to treat this condition is to cut it off. There's no fixing the damage that this does. The lymph system gets granular, it won't expand anymore, and the leg or scrotum or whatever you happen to be dealing with is just permanently deformed. Yeah, it's really ugly. You don't want that either. This is a yeast called Cryptococcus. This used to be only in cancer patients undergoing chemotherapy where their immune system was beat on. Now most of the stuff we see is in HIV patients. Now, the interesting thing here, these are the yeast cells. This is the only encapsulated yeast that is pathogenic. This is it. If you see capsules around here, this is a cytospin prep where it's been spun down from spinal fluid. And this is right stain. This is what we would do when we were initially looking at a spinal fluid to see if there are any cells in there. Here's what they look like as well. That was a kind of a mat of them, so the capsules were kind of butted up against each other. But you can see the individual polysaccharide capsules around here. This is the cell itself.
Yeast.
Here's a better version of it. This actually is a bronchial wash, but it was such a nice presentation that I thought I'd show you. Here's the mother cell, here's the daughter cell, and here's the capsule surrounding it. And you don't want that. Cryptococcus is really hard to treat. That histoplasma I was talking about earlier, this slide may even be back there. Here's the individual yeast. Here's the normal neutrophils, but they phagocytize these yeasts. Normally, you don't see too much of these things in the peripheral blood, but when they start phagocytizing them like this, the load's pretty good. This is a normal, or a nucleated red blood cell, by the way. These are all normal red blood cells out here. Platelet there, platelet there, platelet there, platelet there, platelet. That actually might be a yeast there. That's a yeast cell there that hasn't been phagocytized. We found this about 4 o'clock one afternoon when the CDC came down from the floor. The patient was dead by 3 the next day. This is nasty. This is bacterial meningitis. This is a spinal fluid comes out of the lab to the hematology lab where we do the cell counts and look at it to start with. This is actually, this is gram stain actually, but here's the intracellular gram-negative diptycoxy that you see. This is Neisseria meningitidis. Neisseria gonorrhea looks exactly the same. It does the same thing. They're all intracellular like this. You shouldn't say they're all intracellular. It's not diagnostic until you see them intracellular. But this is the bacterial meningitis that kills a lot of people. A lot of this stuff kills a lot of people. This is spinal fluid from a brain surgery patient that had a traumatic brain injury. And unfortunately, there was a bunch of contaminated stuff in there. And these are all cocci, probably gram-positive cocci that are in here. These are partially destroyed neutrophils. They're degenerate because of what's going on. But that's in spinal fluid. And these are all bacteria that have been phagocytized up to clear them out of the system. You don't want to be that way either.
What do you say?
If anybody's ever had a nasty urine, this is a really nasty urine infection. This came from somebody with an indwelling catheter. They don't change them very often. And once they get contaminated, it can be pretty bad. These are yeast cells, probably Candida albicans, the dark oval things. This one's budding right here. There's gram-positive rods in here. There's some gram-positive cocci. And these are degenerating neutrophils that are trying to clean up the program. I think one of these slides is back there too. I don't know if it's this one. These are uric acid crystals. This is from a patient with gout. You can see that they're kind of hard to see probably from the back, but there's a bunch of needle-like crystals here. I've got a polarized microscope to set up here that you'll be able to see these. This is under normal bright field. You can see them pretty easily. Put a polarizer on them, they look like this. They jump right out. And to figure out whether they're uric acid, or it's gout or pseudo-gout, you put a wave plate on there. I don't remember which way is which. In this particular case, the gout crystals are blue, but if you turn the wave plate 90 degrees, you'll find they're all blue. The ones that notice that they face 90 degrees are more or less 90 degrees off. That's what you're getting there. That it? There. What do you know? That's all you have to suffer. Any questions before we wander through that giant pile of slides back there? Because that's going to be the fun part.
I have a question for you.
It'll be like an hour.
So earlier you showed a deficiency of vitamin D and I think iron. Is that how they find it? They look
Hopefully not. But
I mean, was that really severe vitamin D deficiency, or?
Yeah, for demonstration purposes, yeah, they only use the severe stuff. Because minor stuff, you're not going to start seeing those cell issues. There'll be other problems that show up.
Thank you. And then a general comment. It would be great to end with a picture of a healthy baby or something. I am thoroughly depressed after this. That is
I didn't work with them.
Nobody does.
They went home before I got to it.
unbelievable.
No, that is one of the bad things about working in laboratories. You see a lot of really nasty stuff when you start looking at the slides. Because this is just a little bit. These slides back here, okay, all of the slides, if they're body fluids, some of them are smears, most of them are little circles because we use the cytospin on them. Those you can use at 40x with no trouble because most of those cells are pretty big. You should be able to see what you want to see without going to oil immersion. Leukemias, you should be able to see most of it at 40x. The only stuff that you should have to go to oil on if you want is going to be really the malarias and some things like that. There's a couple bacterial things in here where they're just, I can see them because I know what I'm looking at, and I've been looking at them for a long time, but they're so darn small that in a laboratory I'd go to oil anyway. I was being lazy yesterday.
Did you make all these cells?
All these slides?
yourself?
No, there's a couple in there somebody else made.
Most of them you created.
Yeah, actually, most of these, some of these came from there's some in here that have little paper labels on them that came out of Mercy Hospital in San Diego when I was training in 1975 and 76. And they made sure that we had a teaching set. And I just kinda kept going. If I found something neat, I would, instead of throwing the specimen away, I'd make some more slides, so that I could use it to train my people. And that's what all these were for. was
When I took histology,
And then I went ahead and covered some.
we had to make our own blood smear. We had a cork with a needle through it. And
Okay,
tested
this
with alcohol.
particular punch here is all normal.
That-
Really normal. Just so that you know what you're looking at. Everything else is redelayed. Some of these are surveys. One of the interesting things about doing this.
We had to demonstrate to
is
them
they test you all.
It's gonna take
the
a
white
while.
blood
All.
cells
Years ago, I acquired the library.
under oil
These are
leaves.
survey slides that college